All right, we're going to get started. Thanks for everyone for making it in on noon on a Saturday. Still have half the weekend left. I wanted to pick up where we left off last lecture on the diagram on the left-hand side, because we're going to use this in different iterations when we talk about dietary interventions this morning. The pathway on the left summarizes succinctly the path of glycolysis, so glucose to pyruvate, and then by extension acetyl-CoA, TCA cycle, turning that acetyl-CoA into reducing equivalents, and then oxidative phosphorylation for these purposes is intimately coupled to the reducing equivalents generated by the TCA cycle, so not shown. Amino acids are coming in by those transamination reactions that I talked about last time. In shorthand, we'll show them all feeding into oxaloacetate as one of those products or reagents in those transamination reactions that I talked about before. And where we concluded was where fats feed into metabolism through that process called beta oxidation, giving rise to that glycerol backbone from triglycerides that feeds into glyceraldehyde 3-phosphate, and then the fatty acid subunits getting broken down as acetyl-CoA molecules right down at the nexus here between glycolysis and the TCA cycle. And our goals today are to bring this all together to talk about diets related to obesity control. We'll also talk about how alcohol fits in to this, as well as oxidative stress, something we haven't touched on yet in the course. We're going to go through diets over the decades. First one, I don't know if this is like the 60s or the 70s, was the low-fat diet. So if you want to reduce body fat, eat less fat. This sounds like it should make sense, but there's an alternative interpretation of this now that you have a better handle on how metabolism works. As was discussed last time, all of these pathways from fat breakdown are reversible. None of them are irreversible, like some of the ones that we've talked about in glycolysis or the TCA cycle. Therefore, if fats are removed from the diet, you'll deplete here, but if those calories are substituted with lots of sugars and carbohydrates and other things which will feed in to the top here, it's no problem for beta oxidation or the processes of fat breakdown to run into reverse. You build up more glycerol, you build up an excess of acetyl-CoA, and those are going to start driving fat synthesis. I'd say, okay, forget about fats, just eat nothing. This will work, right? Take away calories entirely. However, there are some consequences to doing that. The first is if you take away all caloric sources entirely, you still do need glucose. The major organ that is very glucose intensive and energy intensive is our brain. Okay, this is why if you starve yourself too much, you pass out, low blood sugar, all those other types of things. And so a lot of our metabolic physiology is geared to ensure that the brain has sufficient energy sources to be able to keep functioning. Therefore, if you take away everything, what that's going to drive your metabolic physiology to do is to provide glucose to the brain to the extent possible via gluconeogenesis. So your liver gets involved. It's going to take other sources and try to drive the synthesis of glucose through the pathways that I talked about before. Now, the key thing here, if you'll recall, when we did the comparison between glycolysis and gluconeogenesis, that last irreversible step from phosphoenol pyruvate to pyruvate, catalyzed by pyruvate kinase, right? That step is irreversible. And so for gluconeogenesis to be able to get around that irreversible step of glycolysis, it needs to go through an oxaloacetate intermediate. So this is where oxaloacetate comes into play when the liver starts driving for gluconeogenesis. By extension, it's pulling oxaloacetate out of the TCA cycle and using it for gluconeogenesis. So you're pulling out from here to drive this pathway up to maintain glucose for the brain. Now you have an oxaloacetate deficiency, and as we said last time, a lot of amino acids are coming in from, are directly coming into or out of the TCA cycle itself. So you deplete oxaloacetate, now you're going to favor muscle breakdown, amino acid breakdown. Muscle is an abundant source of amino acids to maintain that those TCA carbon subunits that are shuttling around the cycle, like I talked about last time. If this is insufficient, then the net result of this also is that if you're driving here, and then you're breaking down fats because you're taking away calories, there's a disposal issue of what happens to the acetyl-CoA. If the TCA cycle is deficient, a lot you're driving through gluconeogenesis. This is getting broken down from fat stores. What happens to these? Is there a question?
Yeah, I was wondering, so the body goes first to like glucose stores, then to fats, and then muscle breakdowns like the last resort, right?
Yeah, so your question relates to the timing of these things. First will be the first depletions are going to happen, actually, they're not shown on the cartoon here, will be those glycogen stores. Okay, so if you have any excess glucose as those polymers, like I talked about a class or two ago, those will be the first reserves to go. You got to deplete those in places. There's a lot stored in your liver, different muscle stores. Those get depleted. Then you're going to start triggering these adaptive processes. One of the foremost ones will be the gluconeogenesis through the literature. And my understanding is that will happen concurrently with fat breakdown. So those are, you can think of as roughly simultaneous processes. And both of them trying to drive glucose. But then what happens to the acetyl-CoA if the TCA cycle is depleted of its intermediates? And then what occurs is that those acetyl-CoA molecules get broken down differently. They can't get fed into the TCA cycle. So what occurs is that they stay, if you go back and look at the slide for beta oxidation, there was a keto intermediate. They'll stay in metabolic intermediate. So these that can travel in the bloodstream, they can be used as an energy source by the brain. And these are called ketone bodies, or simply ketones, as defined by this organic chemistry definition. All right, so we'll have a side chain, as a functional group joined by a carbonyl. Those are that's the definition of a ketone. And these are the things that I don't talk too much about diabetes, but diabetics can be diagnosed by the fragrance of their breath, or their odor. What that odor, the sort of fruity odor, sounds like it smells like they've been drinking, comes from these ketones. Ketones have an odor associated with them, and you can smell that. Some of these things like acetone and are evolved and evaporate from us if you're in a ketotic state. And so there's a lot of different adaptation processes, and this is clearly not sustainable in the long run. I mean, for example, diabetes, which is a cellular starvation response and inability of cells to handle to glucose properly, this leads to long-term damage in places. So this is not, this will work, but is not a lifestyle change that can be possible. A combination of these two, trying to correct that starvation, is the motivation for people who've heard of like the Atkins diet. This is like the 90s, I think, around there, which is starvation with protein supplementation. So trying to remove the driving force for fat storage, favor mobilization, but avoid the depletion of TCA intermediates by feeding with protein. That's what's seeking to accomplish here is feed in here, you can get calories in from here, but largely driving metabolism through the TCA cycle. And this is possible as long as, but it has to be permanent. Okay, so you have to like eating nothing but protein and fats for this to work. And anybody who's done, like had to go on a really low carb diet, my wife had pregnancy-induced diabetes. Once you take those things away, you really crave them afterwards. And it's a strong evolutionary drive to go after sugars and carbohydrates because they're already energy source for your brain. And so some of these things are sort of obvious, right? The thing that's going to work is to simply input and output, right? Fewer calories, in, right? It means that you're not going to have these storage issues. Exercise, what these things do collectively are driving the breakdown of lipids in those fat stores. You also have adaptive changes at the cellular level, that if cells are in habit of really using TCA cycle and breaking down fats and things, you'll actually get more active mitochondria enlargement, and this can also be efficient with fat storage. And then here's where we're going to bring in some of the alcohol intake, and today's the class where I give some personal anecdotes. We're here on a weekend, so I might as well tell some fun stories. I don't do metabolism research, so when I have to teach these things, I make it a little bit interesting for me. And so I said I have 2 daughters, and my older daughter, she was asking me, this was a number of years ago, but she's like, Daddy, I want to have one of these vitamin waters for my lunch. I'm like, don't have vitamin waters. They have like, what do they have? 140 or something calories. And I'm 125 calories. Don't drink any of those things that you don't want. It's just sugar. She's like, well, why do you have them then? I said, okay, you've got a good point. So let's do an experiment. I'm going to have a dietary intervention in me. I'm going to take away this sugary drink that I just reflexively would have every day at lunch, and then see what happens to my weight, because I just took away 125 or whatever calories out of my daily intake. And you're going to record the data with me, so we go and we do the thing. So I take a baseline reading here, and you can see there's ups and downs. Everybody has fluctuations. And then we were going to have the intervention. So I stopped my vitamin water at this time, and then I was going to see what occurred afterwards. And so we're watching. So there's a little bit, thank you. Well, there's a little bit downward trend. I'm not finished, because there's more stories to tell. You see there's some downward motion. Nothing really all that remarkable. Maybe the intervention was not strong enough. But what I was struck by is the real ebbs and flows on a day-to-day basis. And then I realized that we had other activities. In fact, I had 2 weddings at this time. And so I must have liked the open bar better than the dance floor on those days, because you can see the spikes that came up immediately the day after. And so how does that relate to fat, weight gain, all these other types of things? And this goes back to those intermediates that I had talked about between the output of glycolysis and the input of the TCA cycle. Recall that we handled pyruvate differently under anaerobic conditions, under fermentation conditions, than do yeast. But when we intake alcohol, it starts feeding into those yeast-like fermentation processes, but in reverse. So if we consume ethanol, we have the alcohol dehydrogenase. Now there's more reactants here, giving rise to the next product, which would ordinarily run in the reverse during alcoholic fermentation. So there's alcohol dehydrogenase. We have an intermediate here, which is more to be said about that. And then this then eventually gets processed by another dehydrogenase called acetaldehyde dehydrogenase to yield acetyl-CoA. So acetaldehyde is the next byproduct downstream of ethanol in us, running that alcohol dehydrogenase in reverse. We generate another reducing equivalent with the second acetaldehyde dehydrogenase to yield acetyl-CoA. And now we have an abundance of acetyl-CoA, which is at that bottom part right there, or the top of the TCA cycle, or reciprocally the building block for fat storage. And so the immediacy of ethanol intake to building blocks for fat synthesis is why you can get these partly the reason why you can get these rapid swings in fat storage as a result of ethanol consumption. This pathway is also why we get sick with excessive ethanol consumption, because this intermediate here is not normally in us. We don't have that, the pyruvate decarboxylase that would ordinarily handle that intermediate in yeast. Recall that's the reason why we can't get into this pathway. And so this is the thing that will cause us problems. It's a reactive group. That aldehyde will perform chemical reactions on biomolecules. We'll speak more about free radicals in the mitochondria in a moment. but also by virtue of these chemical reactions, it can deplete things like antioxidants, the things that ordinarily protect us from the free radicals. There'll be more to come on that in a couple of slides. And then because of the chemical reactivity of these acetaldehyde groups, they can chemically modify biomolecules, forming so-called adducts, so chemical appendages there of this acetyl group onto places that they ordinarily wouldn't be. These can be antigenic, so neoantigens, new epitopes that ordinarily don't exist on us, generating inflammation, immune type of responses. And by virtue of that inflammation and the immune cell responses, this can trigger ECM secretion, collagens and things. Professor Barker, like he had told you in the first half of the course. And a lot of this is happening in the liver. Key metabolic enzyme, this is a lot of the ones that's handling the ethanol burden in us. And these are the hallmarks of organ change, notably cirrhosis, the fattening, hardening, and fibrosis of the liver that occurs if in alcoholics and extreme drinking over long periods of time. A couple other things to say about on this slide. I'm trying to like Asians in here can't drink, can drink. In the Asian population, there are two alleles that apparently my wife has both. One is a deficient allele in acetaldehyde dehydrogenase. And so if you've ever gone out to a bar or somebody who has that allele, these are the individuals that get flushed and red when they drink because they can't handle the acetaldehyde as efficiently as others, and this gives rise to some of this inflammation, flushing. Right? That's a result of deficiency there. There are also people who get no enjoyment out of drinking, and there's actually a hyperactive alcohol dehydrogenase that rapidly processes ethanol into acetaldehyde. And so what happens when my wife, well, she doesn't touch anything, but if she were to, she feels nothing but the acetaldehyde. So she goes straight to red and will throw up. She'll get sick very quickly because of some of these differences in the population. It's a lot hotter in here than it is in the base of pin. So give me a second. Okay, so I'm going to talk about one more intervention that is a newer hybrid. People maybe have heard it, not heard about it before, and it's this idea of intermittent fasting. So starvation, like metabolism, like I talked about before, but doing it every once in a while, but on a weekly basis. And so what's the rationale for this? You get those starvation-like responses. Okay, so you're going to deplete the glucose, glycogen stores. You're going to drive the TCA cycle, metabolic intermediates, fat mobilization, striving for a mild state of ketosis. Okay, so you're going to deplete this, but not for a long period of time, but enough to really deplete all of the intermediates in the pathway. You do that for a day, so the 5-2 stands for 2 days like this, and then 5 days just regular, like normal caloric intake. And the thinking is that you've depleted everything, there have been some adaptations, and then when you go back to eating, most of the calories the next time through are not dedicated to fat storage, they're dedicated to filling up all the things that you've depleted in the day before it. So of course, if I teach it, I have to try it, right? So this was my experiment 2 or 3 years ago. And so I was doing it during class, and so it was sort of interesting to see. By frame of reference here on the right-hand side, this was my vitamin water range. So this is where I was starting, a couple of baseline measures. And then on these starvation days, you're supposed to take like 600 calories total. So by virtue of, by frame of reference, you're supposed to have a 2,000 calorie intake day. So it's like 1/3 roughly of your intake. And so that was where I was a couple of years ago, and then I extended the measures here. Yeah. I wasn't sure when I was going to stop, but then I figured to stop about here, so I'm about at that point. So that's me. I eat about 1 day less per week than everybody else by virtue of this. How is this different than eating less overall? Yeah, it is not. If you simply decreased the calories and you averaged it out over that time, it helps more with the mentality of you have 1 day, you make it through the next day, and you can eat the following day, as opposed to you're always hungry every single day for the rest of your life. So it's like feast and famine. And so, I don't know, for me, you get older, you get a little bit bored, and so this shakes up the week every time. So. These classes are my eating days, just for frame of reference. I teach a little bit different on the starvation days. Okay. When we talked a little bit on the acid-aldehyde intermediates, we talked about free radicals and oxidative stress, just mentioned it. I want to talk a bit more in detail now about this, and this circles back to oxidative phosphorylation that we touched on last time. Oxidative stress is referring to, we've seen this back and forth before. Things are getting oxidized, things are getting reduced. As long as there are electron donors and acceptors to be able to handle those things, in principle, everything is fine. But if one of these electrons were to leak out and be released, this is how you can get this imbalance that will create reactive intermediates that can go on and do secondary damage in the cells. And these oxidizing compounds fall into two main categories. The first one are reactive oxygen species, or ROS. The two main players here are superoxide and peroxide. I give the chemical structures here, so gaining 1 electron or 2. And there are also reactive nitrogen species. One of them is normal and short-lived. You'll recall nitric oxide as a second messenger, so that is a reactive nitrogen species. The one that's more damaging is if nitric oxide comes into contact with a superoxide, thereby generating a peroxide generating a peroxynitrite. This one is problematic from a cellular damage perspective. And what I want you to take home is that even though we said that there are these huge metabolic advantages of aerobic respiration from the perspective of converting glucose into ATP equivalents, this is also the place where reactive oxygen species are generated. We're going to zoom in a little bit on complex 1, but the processes that we're talking about happen in other places along the electron transport chain. And I don't have this slide here, but if you'll remember the crystal structure slide that I showed where the electrons are coming off NADH, and there's that first acceptor of those electrons, flavin mononucleotide on complex 1. This is another coenzyme that can undergo reduction and oxidation. This is the major leak source off of complex 1. So that handoff from NADH to flavin mononucleotide happens, but doesn't happen with perfect efficiency. And so as a result of that reduction, can leak off an electron. You're under aerobic conditions. Oxygen is in the environment, and now you're generating reactive oxygen species. These types of leak processes also occur in complex 3, but the leak is more severe in complex 1. Yes?
So the production of ROS only happens with aerobic respiration?
Are reactive oxygen species only generated during aerobic respiration? No, there are other dedicated peroxidases and activation processes like the ones that come to mind, even separate of metabolism in general, like the one that immediately comes to mind, like neutrophils, for example. They mobilize NADPH oxidases to fight bacterial infections. Those generate a lot of reactive oxygen species because you're trying to kill the bacteria. Those are things that are in the vicinity. And then aside from more specialized cells, some of those oxidases can be leaking out, but definitely the mitochondrial metabolism is a major source, but not 100% source. When you have superoxide generation, the types of things that can occur are, one of them is carbonylation on arginines. Here's an example. So you have superoxide tackle one of these arginines, and now you're getting a formaldehyde group here on the end, which is, an aldehyde group, which has the same chemical reactivity like acetaldehyde. That's not a good thing. One of the places where free radicals are especially dangerous are places where you have double bonds, because the electron comes, attacks the double bond, breaks the double bond, and then frees up another radical to go on and do another reaction. And so one of the places where this comes to bear very prominently is in lipids. If you have lipid peroxidation, unsaturated fatty acids, they have those double bonds, I'll call from the membranes lecture. Those can set this cascade of lipid peroxidation in the membrane, because all of those double bonded lipids are right there. And then finally, DNA is also damaged. I tried to look into this to tell you very cleanly what's the modification that occurs on DNA. It's not clean at all. There's a whole spectrum of lesions that are generated as a result of oxidative stress on DNA. But I can tell you that they're all single base lesions and are repaired by the BER, the base excision repair machinery, just like you had learned about a couple of weeks ago. Because we've had a long time with our mitochondria, we have evolved mechanisms to handle this. I don't want you to get the impression that every single superoxide is trashing some aspect of our cells. And the reason is because we have a whole series of pathways to capture those reactive oxygen species and, if you will, absorb those free electrons so that they don't damage other biomolecules. And those pathways are catalyzed by what are called scavenger enzymes, and they revolve around this biomolecule called glutathione, or GSH for short, which serves as an antioxidant role inside the cells. People are familiar with antioxidants. Just generally, this is one that we produce naturally because of its involvement in these pathways. Let's start with oxygen. We have leaked an electron off of complex 1, and now we have superoxide. One of the scavenger enzymes for superoxide is an enzyme called superoxide dismutase, or simply SOD, that will convert superoxide into hydrogen peroxide. We then have another enzyme called catalase that will turn peroxide into water and oxygen, enabling the superoxide to be handled into more palatable intermediates. The place where glutathione comes into play is that depending on the state of the cell, the abundance of these enzymes to be able to perform these two reactions, glutathione, whose chemical structure is shown here, can also suck up the free radicals in these intermediates. And the way that those free radicals are sucked up is through the thiol group here. That's why it's called glutathione. There's the thiol group that can undergo reversible oxidation and reduction. So we have two free glutathiones with a glutathione peroxidase. In the presence of hydrogen peroxide, we'll take those two free thiols and create a disulfide bond, so GSS, so disulfide bond here, that then can act as a buffer, if you will, of those free electrons until they can get handled afterwards. So if there's a burst of reactive oxygen species, they'll go into these disulfide bridged glutathione molecules to protect other biomolecules from getting irreversibly damaged by the free radical process. This disulfide can go back and forth. Generally, oxidative stress impinges on almost every facet of biology, but we often seek to avoid studying it because it's very hard, even though my lab has some work that's doing it. Because if you generate one of these reactive intermediates, they're short-lived. They react with pretty much every biomolecule. They can run through these different cascades and become other reactive species along the way. And then you have all of these different adaptive responses that seek to quell the impact of the reactive oxygen species. And the last piece of dirty laundry on oxidative stress research is that there's a lot of context specificity to these, including how you run your experiments and the different environmental conditions that you use to study these pathways. And so it's a very complicated field of research that has a long history associated with it, but it's kind of its own thing because it's very unique in biology. Yeah. Sure, you're asking about dietary antioxidants, vitamin C, ascorbate, or vitamin E. A lot of these, yes. And so these are dietary sources that can come in, and then they simply have the what is an antioxidant, basically one that's able to accept electrons as their free electrons as they're generated by these reactive oxygen species. The idea being that you ingest them, you elevate your circulating concentrations of them. If you have oxidative bursts occurring metabolically, that some of them will get sucked up by those external antioxidants. Because they don't even necessarily need to be enzyme catalyzed processes. You just have them there, and then they'll convert those free radicals. They'll get chemically modified into an inert substance, and then that's sort of the end of it. There were two questions in the front here. Are nitrogen radicals not as harmful? I'm going to go out on a limb here. I'm not 100% sure because this is not my area. I think that the nitrogen radicals are even more dangerous than the reactive oxygen species. And the reason is I'm not aware that all of that handling with superoxide dismutase and catalase and the glutathione peroxidase, that's for superoxide. Reactive nitrogen species don't have a similar adaptive mechanism. Okay, and so if those come together, pretty much they're going to modify something inside the cells. Unless some of these dietary antioxidants may be able to suck them up. If you look at the chemical structures on them, they're going to have some type of double bond that can accommodate the free electrons. There was anyone else here? No? All right, that's it with metabolism. The two main people I think of for, if you're interested in metabolic research, are Thurl Harris in pharmacology, and Zhen Yan is more exercise physiology and mitochondrial physiology in medicine, and also in the cardiovascular research center here on this floor. Questions, high level on metabolism? All right. Mm-hmm.
A quick question about the alcohol metabolism. So we were talking about people who have hyperactive NAD+ production to make a lot of acetaldehyde from ethanol.
Let me just clarify that one part. When we're talking about differences in the population, alleles of being able to handle ethanol, it's not at the level of the availability of NAD coenzyme, it's the enzyme activity itself. So the alcohol dehydrogenase has you have different alleles that have different catalytic efficiencies, or even to the extent of being broken that will handle those differently.
Okay, so I guess let me reword. So
Mhm.
for the people that have the allele for hyperactivity of the alcohol
Mhm.
gluconeogenesis, to make it into acetaldehyde quicker, does that affect like tolerance of people? Is that how that's determined, or?
Yeah, so that first step, the alcohol dehydrogenase, if you're hyperactive, how does that relate to tolerance of alcohol and things? Yeah, it does, because the thinking, I think there's still some debates about exactly why ethanol gives people the feeling that it does. Last I heard some thinking about membrane fluidity and things like this. But if the enzyme is taking that away, yeah, you're not going to feel anything. So that's why, like, my wife doesn't get any, never. It just goes straight to sickness. And I know other friends of hers that if you have like that allele and then not the other one, there are some Asians who I've been out with that seem like they can go forever. So yes, there was other questions. Yes.
I was just wondering, because I've heard of this, like the diet called the ketosis diet, where like you're purposely in ketosis all the time. Is that like, yeah, I just
Yep.
know like the gym I was at this time, the head guy was always like, oh, ketosis, he raves about it. And
Yep.
I was wondering like if you know more about it or.
Yeah, the other thing on that, all that the keto stuff is related to variants of this theme. Okay, what it means is you're trying to get all those sugar stores depleted in your body to try to mobilize these other fat stores. And the challenge, I think, of doing it, I'm not a dietitian, okay? I just teach this stuff. But the challenge long term, what you turn to are like diabetics, right, who are chronically in a ketoacidotic state if they're left untreated. Your brain can do okay with those as energy sources, but other parts of your body long term don't like it at all. Diabetic foot ulcers, retinopathy, so your fine vasculature and things seems to be very susceptible to prolonged glucose, prolonged deficiencies in circulating, that's a prolonged elevation of glucose that you can't handle. But as a result of not being able to get the right metabolism, you're having secondary effects on that. So what the debates are now is like, how long do you go into that state and how long that is good for you? There's other cellular processes that we don't have time to talk about in this class, but the one that comes to mind is autophagy, which means like when your cells are starved from one another, you start digesting some of your organelles, like your mitochondria, and that actually might be a good thing to do periodically, to simply recycle the old stuff and get it and use it as energy sources for a while to turn it over periodically. And yeah, but they're all seeking to try to do that same thing. All right, what I'd like to do, I have a target for this, is make a shift now. We've covered metabolism, and what we're going to conclude the class with is the cell cycle control in the setting of cancer. And the reason why we position this lecture where we do is that it's a nice closing foray for the course, because it enables us to integrate many of the other topics that we've covered so far. Today I'm going to set the stage of what we're going to dive into more deeply on Tuesday and introduce some of the vocabulary that's particular to cancer, because my lab does cancer research. I'm going to start using some of those terms without thinking about it, so I'm going to walk you through them. Cancer remains a very big problem. Okay, 1 in 4 deaths, lots of money spent in the healthcare system. Most of the cancers arise from random mutagenic events as a result of our environment, as a result of errors in DNA replication, as a result of chronic inflammation. I was talking about some processes that can give rise to reactive oxygen species with chronic inflammation. Only a very small fraction have been tied directly to viral infections, but that's the one we're going to talk about. And so the cancer type that has, say, the strongest link with a viral promotion and viral origin is cervical cancer related to a category of viruses called human papilloma virus, or HPV. There are many different strains of HPV that can promote anything from benign type of lesions, warts. Warts are hyperproliferative lesions on the skin, not cancer, but it's a disruption of normal cell cycle control. There are also strains that are associated with cervical cancer, and I'll talk about the ways in which that, and we'll be able to tie this in to some of the disruptions in cell cycle control at the end of class next time. HPV is associated with cervical cancer, and it's also associated with head and neck cancers. There's a couple things about HPV. Hopefully that will not freak you out too much, but it is very prevalent, okay, in the population at large. It is a transmitted disease, sexually transmitted, mucosally transmitted disease, and we show the age-related prevalences here. I have this number, the prevalence is about 20% and 27% in women overall. There was a study in 2017 that now the prevalence of HPV, oral HPV infection in guys is surpassing females now. And this spike coincides with overall activity, if you will, in the population. And so something to be aware of, it's out there in the population at large, but being positive for HPV infection is not a guarantee for cervical cancer, but all instances of cervical cancer will have HPV infections with them, virtually all. And I can't remember what the percentage is for head and neck cancers. I think it's probably about 30 to 40%, if I remember the studies on it. Yes? Oh yeah, what is head and neck cancer? You can think of it as any tumors arising in this area that can't be ascribed to like a leukemia or a lymphoma. So they're like in the esophagus, up in the mouth, in the back of the mouth area, it's like that. But it's a bit of a catch-all for anything that arises in that area. Mm-hmm. Yeah. Yep. Oh, that's the I said before. Let's talk about cervical cancer specifically. The incidences are shown here on the top. You can see how many the cases. I don't need to read this off the slide. The way that these are diagnosed, this is a gynecologic visits, things like pap smears, which are trying to take a sample of the cervical epithelia to be able to assess how normal or abnormal those cells look upon sampling some of those. Right. This is what a cervical imaging will look like, and all of this white region here, this is the cervical cancer spreading across the epithelium. Inflammation, that's what the blood and the pus and things are. For cancer overall, there's basically an entirely, it's of its own vocabulary, and some of these things you just need to know to be able to keep abreast of literature and things in the field. And the definitions of some of these terms are embedded in one another. I'm just going to walk through several of these. One of the major distinctions in cancer is whether or not a particular lesion that looks abnormal or atypical is benign or malignant. And so the benign lesions would be like a wart, for example, or a mole. We talked about how nevi on the skin can pick up a mutation, proliferate for a while, and then stop by senescence. Those would be considered benign lesions. It's the malignant ones that are of special concern, and what distinguishes them from benign lesions are three characteristics. Anaplasia, which is defined as loss of differentiation. 90% of cancers arise from epithelial cells, and the tumors that arise from them are called carcinomas. Epithelia are differentiated cells, polarization, all these things you've seen from before. The loss of that organization, the loss of epithelial characteristics in carcinomas is anaplasia. So looking differently, losing the specialized function of the cell of origin is one characteristic of malignancy. The second is invasion. Cells occupy particular parts of our body. They have a certain arrangement that's characteristic of their normal architecture. When that architecture gets disrupted and cells go into places of the tissue that they're not supposed to, that's invasion. This is locally. Metastasis is a more widespread relocalization of the cells. This can be to the lymphatics, in the bloodstream to an entirely different organ. And so that dissemination of the cells from the primary site to distant organ is metastasis. These processes of malignancy, loss of differentiation, invasion, metastasis are thought to accrue sequentially. And that process of accumulation of mutations, accumulation of changes, and the progression towards malignancy is called tumorigenesis or oncogenesis. I'll give an example of anaplasia and invasion in breast cancer, which is what my lab works on. This is a cross-section image of normal breast tissue stained for a collagen molecule shown on the bottom here. So normal breast tissue has ducts and lobules, so the tubes, milk-secreting conduits out toward the nipple, and then lobules, which are grape-like sacs of breast epithelial cells that are the milk-secreting units of the tissue. And so what's shown here is a cross-section of a couple of these alveoli and ducts, so the spheres, and then the conduits. Nice polarized epithelia all along the side here. This collagen expression is localized to the luminal epithelial cells all along here. And so now, same stain in a breast cancer patient. The collagen is all over the place. This is all extracellular matrix. It's not positive for this collagen, but you can tell by the histology about what it is. Here you can see this invasive finger here of cells. These are cancer cells going into the tissue. And then the other thing, I'd like you to appreciate all of these cancer cells here. You can see large nuclei with diffuse blue staining. That is indicative of this, what distinguishes a cancer cell from normal cells here. These are at the same magnification. A couple more terms. If we are talking about solid tumors, they'll often begin as a mass, right? So a hyperproliferative mass locally. Right? They haven't invaded yet, but they've escaped the normal growth controls, so they're going to start growing. Once that mass has grown above about 1 millimeter, diffusion of oxygen is no longer sufficient. They're going to keep growing. They've lost those normal proliferation controls of normal cells, but now the cells in the center are going to become hypoxic, deprived of oxygen, and they will start dying. But as this tumor continues to grow, you're also selecting for processes to try to provide more oxygenation of the tumor as it's growing. And that process of recruiting blood vessels, growing new blood vessels into the tumor, is called angiogenesis. Talked about oncogenesis, so oncogenic, tumorigenic, and oncogene. We need to get down to oncogene a little bit. So oncogenic simply means does it cause cancer, not tumorigenic synonyms. Oncogene is a more particular definition. So an oncogene the most careful way I've been able to define it is a gene whose product causes cancer. And what would that mean? That would mean that that is a gene that can, in a person, has been documented to be a causative event in tumorigenesis. A lot of times, oncogenes are disrupted forms of a normal gene in our body. That normal gene is called a proto-oncogene. Try to give some more context to that. Let's say that we have a kinase in us that's important for proliferation generally. You'll see a couple of these in the coming slides. If there's a mutation in that kinase that all of a sudden turns it on all the time, now you have a driving force to proliferate, proliferate, proliferate without any of the breaks in the simplest scenario. So the normal form of that kinase would be considered a proto-oncogene, whereas the mutated form of that kinase would be an oncogene. Yes, I see a hand.
Is that like, are those the genes that get mutated when you're in the sun too long and you get some cancer?
Are these the genes that get mutated when you're in the sun too long? Some of them are, yeah, and in fact, if I can, I'll try to remember precisely the pathway that, you remember we did the MAP kinase pathway, those RAF, MEK, ERK. So RAF, there's a very characteristic mutation there that's gained in melanomas, and it's so well recognized that we have a mutated RAF specific inhibitor that people use to treat melanoma. So in that specific example, RAF is the proto-oncogene. The mutated RAF is the oncogene.
For the cells that escape growth control mechanisms, are the donor cells that they make also not subject to any controls?
Yeah, and so your question is, if you escape those growth controls and then you're starting to proliferate, 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 do those rules still apply to all of the progeny of that original event? Yeah, so cancer is a disease of the genome, at least at this most fundamental level. So if this is an oncogene-driven event, every descendant of that cell is going to maintain those mutations and pick up new ones.
So then is there now a cancer that routinely spreads without being oncogene?
Is there, can you rephrase that?
So if it's a mutation in the DNA and it's caused by the oncogenes, are all cancers that proliferate like this oncogene products?
Oh, yes, I see what you're saying. So your question is there always an oncogene driving a cancer? Yes. But there are, and in fact, there are examples. The Cancer Center seminar speaker just yesterday was talking about, there are examples, though, of cancers being driven by one mutation. So, or one event, one oncogene, if it's really potent, can be sufficient to drive certain certain cancers. Oh, now I'm thinking, let me get back to this because there's one more thing I need to talk about. You're right, so this is the proto-oncogenes. There's a caveat to that, I should say. Because you have the oncogenes driving tumorigenesis. The antonym of those, or another category of genes that inhibit cancer, called tumor suppressor genes. So tumor suppressor genes, you'll see some of them in the next lecture. They help enforce those proliferative controls on cells. And so there are examples. The one that comes to mind is retinoblastoma. There are examples of some childhood cancers that are driven by a single loss of a tumor suppressor. So there is always a lesion, but not necessarily in an oncogene. You could break a tumor suppressor or gain an oncogene. Usually, depending on the cancer, we now have a pretty good understanding of the mutational burden on the typical cancer of that type. So diseases like leukemias and lymphomas, I'm trying to drop some names of things that you've heard about already in the class. Those are usually pretty low on the mutational burden, early onset, things like that, relatively simple tumors. If you want to talk about skin cancers, lung cancers that have a lot of environmentally driven mutational events, those tend to be the heavier mutational burden. Those could have hundreds, many hundreds of mutations in them. some of which are driving the tumor, some are just coming along for the ride, but that tends to be the typical burden. So you can go anywhere from 1 to several hundred. And then one other thing, sorry, now I'm in my area, so now I tell you all sorts of stuff. There was also just there've been a recent set of studies where the oncogene driving events some of them are going to be necessary, but it doesn't guarantee that they're sufficient. So like in our skin, as an example, we will have fields of cells that have lost a tumor suppressor or picked up something that we would have thought would be ordinarily oncogenic, but they've not escaped any of the other growth controls. There's still enough pathways holding those in check, so it's not cancer. And so it's getting comfortable with this idea that our cells, the somatic genomes of our cells are evolving over our lifetime as a result of environmental exposures and random chance just during proliferation. Oh, that's a good segue for this next slide. All right, so these are time-dependent processes, especially those that have the tissues that have a constant environmental exposure, like our skin, or constantly undergo rounds of proliferation, like our gut. Those are the ones that will have this age-dependent, or all cancers have an age-dependent increase, but this is the thought to be the origin. Why this is because it's simply a matter of time, random chances, where will mutations land in genes versus in non-coding regions or other places that may be a bit safer. And just because that there's a mutation picked up in a tissue, that doesn't mean that it's oncogenic. But as time goes on, the incidence increases. And I'll note that this is a log-log plot, so that these straight lines here are exponential increases with age. Your questions already preempted this slide, but what I want to say in a little bit more completeness is that a lot of these oncogenes or tumor suppressors are bearing down directly on the control of cell proliferation. And these are at many levels that you've already been introduced to at different points in this course. They can start at the level of growth factors. Right, so these are diffusible protein hormones that create a external stimulus to go into the cell cycle. These can be dysregulated. Those growth factors have receptors, transmembrane receptors on the plasma membrane surface. They can be amplified. You can gain many copies of them to cause them to be more sensitive to a certain amount of growth factor. They can become mutated to become active even in the absence of ligand. There are also a number of intracellular signaling proteins. We just talked about RAF by virtue of the question, but there's other ones all along different proliferation pathways. And this continues to go down into central dogma. All right, so there are examples of mutated transcription factors, both oncogenic and tumor suppressive transcription factors that can be disrupted, as well as other things. We're going to not talk too much about the anti-apoptotic proteins, but we will talk about cell cycle control and how that can be dysregulated, as well as DNA repair processes and how those, there are a number of important tumor suppressors in the pathways that repair our DNA. I want to take one slide from the pre-lecture to say a couple things and set the stage for what we'll do in next class. First, to remind that you can break down the cell cycle in two main levels, the M phase that encompasses both mitosis and cytokinesis, as well as this interphase where the DNA is synthesized and proteins, other biomolecules, are doubled to prepare for a round of mitosis and cytokinesis. We have S, which is the phase in which DNA synthesis occurs, flanked by two gap phases, G1 and G2, where the cell is growing. All right, so ribosomes are doubling, organelles are doubling. And then the last phase, this cul-de-sac over here, is the quiescent state, a resting state that people often call G0, which means exited from the cell cycle, but can come back in if there's an appropriate stimulus. Senescence, one of those ones that we talked about before, that's like a dead end out from there. You kind of exit, never do you go back in the cell cycle. we go around this cycle, there's a couple of things that we're going to talk about in much greater detail next time. There's two main ways in which that cell cycle is regulated or controlled. The first one relates to the timing. How long do you last in S phase? What are the things that dictate transitions from one phase to the next? And these are regulated by a category of proteins that ebb and flow. Their protein abundance increases and decreases as a function of where the cell is in the cell cycle. More to come on that. Overlaid on those cycling proteins in the cell cycle, there are what are called checkpoints, which are stops on that normal cycle if the conditions are not right for the cell to proceed to the next phase. They're schematized here. First is the G1 to S transition. We're going to talk a lot about that in detail. What that is evaluating is if the environmental conditions are right to go on and undergo a round of doubling. But there are others which I want to mention here for completeness. The second part here, the G2 checkpoint, if you're going to divide the cell, you want to make sure that every other aspect beyond the DNA has been appropriately doubled. If you think about this, if you're off by 5% in your cell mass, your cell volumes, things, you've either grown or shrunk by 2.5% once you divide the cell. And if that's not corrected, the cell's either going to blow up or will shrink down to nothing. And so cell size control is a still mysterious facet of biology. People don't exactly know how the cell can evaluate it, whether it has the right doubling of everything else. And then the final checkpoint is called the spindle assembly checkpoint. This relates to the alignment of proper alignment of chromosomes during metaphase before the anaphase and segregation of the chromosome. If those sister chromatids are not perfectly lined up, joined by myrotelic attachments by the spindle via the kinetochore. I'm pulling out a whole bunch of stuff that you heard early part. You're going to pull both sister chromatids to one of the cell or the other cell. This is a really important consideration for chromosome stability and the avoidance of aneuploidy. We talked about that term before. Thus, next class, our goal is to fill in the gaps here, the question marks, from how a cell, when it receives a growth factor stimulus from the extracellular environment, processes that information intracellularly at the level of cell cycle regulation and the G1 to S transition to go in and replicate DNA at S phase and then undergo a round of mitosis during M phase. Questions? All right, I hit my mark 5 minutes early. I'll see you on Tuesday for the grand finale.